Yes, okay, so let's start our questions and answers about embryology. So, proper introduction of our speaker today. This is ours, Olga Chapya, senior embryologist at IVMed Embryology Laboratory, ASHRAE certified senior embryologist, and she has 11 years of IVF and genetics laboratories practice. And she has kindly agreed to be our speaker today. So let's start with the questions. The first one is, I'm 35 years old. My husband is 40. We did PGD9 and transferred embryos several times. We had many negative results and miscarriages. I've heard that PGD should decrease the miscarriage, but I still have it. Maybe we need to do NGS. What is your opinion, Olga?
Well, to be honest, it's a very difficult situation, and so the question is also difficult, and it's really difficult to answer it plainly, but I will try to make it at least simple to give an adequate answer. So first of all, when we talk about PGD, in population, generally, on a big amount of cases, PGD really decreases the number of spontaneous abortions. It is a proven fact, and it's quite logical because PGD help us to identify healthy embryos. That's why we avoid the possibility to transfer unhealthy embryo into uterus and get pathologic pregnancy. But it is in population, and when we talk about any individual situation, we should think about it individually. So in your particular case, I'd like to remind you that pregnancy, its occurrence and its outcome is actually a cooperation of maternal and embryologic factors. That's why, first of all, we have to think what may be the reason of repeated implantation failure and miscarriage in your case. That would be great if you had cytogenetic testing of abortion material from your previous miscarriage. It will help us to know whether the embryo was healthy or not, because it is widely known that it's a very common reason of miscarriage, that the embryo is not healthy. It may have some chromosomal abnormalities, and of course they lead to the termination of pregnancy at earlier stage of its development. But from the other point of view, if the embryo was not tested with cytogenetic tests, it doesn't mean that it, of course, was unhealthy. So we have to keep our eyes on female health too. So that's why I strongly recommend you to find a good gynecologist who can exclude any other factors from maternal side. It may be some structural abnormalities of uterus. It may be some immune problems, for example, antiphospholipid syndrome or abnormal reaction of native killer cells. Some hormonal problems, influence of your lifestyle. There may be plenty of reasons, and we should exclude them before we say that the embryo is the reason of such a sad situation. But apart from that, yes, you're definitely right. When we talk about PGD9, we know that we have a limited information about embryo health because we test only 9 pairs of chromosomes, while embryo has 23. That's why after consultation with a reproductologist who will test your health, who will think about your situation very thoroughly, you may go on for the second attempt and then do NGS testing in order to avoid transferring of abnormal embryo. And of course, before going for subsequent IVF cycles, please do cytogenetic study, so-called chromosome passport sometimes, because in order to go for a good PGD, we need to know that you and your husband are healthy by themselves. So good luck to you, and I hope that your case will end successfully. Thank you.
Thank you. I'm sure the couple has heard the answer that they were hoping for. The next question is, If embryo stopped its development on the third day, the issue is the sperm. Is it true? My husband is 30. He doesn't smoke, has healthy lifestyle, but our embryo stopped the third day 2 times. What is your opinion on that?
Well, no. Sometimes it is speculated that if embryo stops at day 3, it means that the reason is husband. Definitely no. The other thing that we are sure today is the fact that from the third day of development, embryo switches on its own genetic material. It means that at the day of fertilization and second day of its development, embryo develops due to the oocytes' help. It means it uses the energy of oocyte, the regulatory molecules of oocyte, proteins, RNA, DNA, everything. But when it comes to switching on embryos genetics, sometimes trouble happens. And of course, we don't know who is the reason of the trouble, whether spermatozoa or maybe oocyte. Or maybe their combination that just gave I would say, unhealthy result, unhealthy embryo. Probably the highest, I would say, the highest opportunity, the highest possibility how we can explain such situation, especially taking into consideration that it's repeated, is the chromosomal abnormalities of embryos, because they are very frequent when we talk about pre-implantation embryo development. And usually, in such cases, we recommend PGT, so testing of embryos to be sure that they are healthy, but in your particular in your particular case, you're not able to test embryos because they just stop their development. The other reason for such a situation may be so-called energetic abnormal supply. We have so-called mitochondria. It's very small structures located at every cell of our organism that give us energy for development and for the cell dividing. But sometimes their work is not sufficient for keeping up embryo development. And in such situation, we can recommend you a novel technique of pronuclear transfer that will help us to replace the cytoplasm with mitochondria, but leave your genetic material to develop further. And of course, as a diagnostic, it's really difficult to say in such situation, but as also an option for diagnostic, we can discuss so-called separated cycle, and half of the oocytes, for example, are fertilized with donor semen, and half of the, and also we use donor oocytes that are fertilized with husband semen. And afterwards, we control the development of embryos, of each embryo. And afterwards, we can see what is the reason, spermatozoa, oocyte, or maybe their combination. And usually afterwards, we will get at least developed embryos for the transfer. But still, it is a very, very difficult step to go on for such a testing. That's why probably you should consider visiting doctor, talking to doctor to consider your particular situation and to find the way maybe not to go on for such a diagnostic IVF cycle, but to select any other variant for treatment. Maybe there is another way. Thank you.
Thank you, Olga. So you should be careful not to blame everything on sperm, right?
Yes.
Can I transfer mosaic embryo? Are there any risks?
Yes, of course. You have the right to transfer mosaic embryo. But before you go for such an important step, please talk to your geneticist. Because before you go on for such a transfer, you have to understand all the risks properly. So the doctor will explain you the details about this particular mosaic embryo you're going to transfer. Because the prognosis is totally different for mosaic embryo depending on what chromosome is involved in mosaicism and what is extent of mosaicism, because there are different types. It may be severe one that is higher than 50% and maybe moderate, less than 50%. And of course, the prognosis is totally different. But what we can say confidently for today, First, mosaic embryos can give healthy pregnancy. It's for sure, and we know it definitely according to the results of numerous mosaic embryos transfers made in the world. Second, mosaic embryos for sure have limited ability to implant. If you compare them to healthy embryos, well, they implant twice less compared to them. Of course, it should be noted that in a minority of cases, mosaic embryo can also implant and form a pathologic pregnancy with a mosaic or even totally unhealthy fetus. That's why after transferring of mosaic embryos and pregnancy occurrence, you have to consult your geneticist thoroughly and to probably to go for pregnancy testing, like invasive testing, like amniocentesis. It's done on 20th week of pregnancy, or maybe it would be enough to go for non-invasive blood test. But still, it's a very important issue when we transfer mosaic embryos. We always keep in mind that they have a little bit higher probability to give a pathologic pregnancy. So please be attentive, talk to your doctor, and good luck.
Thank you, Olga. Let's take a look at the next one. I'm 29 and I have lupus. I'm taking the medicine prednisolone. Can it affect egg quality or quantity?
Saying honestly, we don't know exactly to what extent prednisolone can affect the quality and quantity of the eggs of a particular patient. Just to calm you, I would like to mention that sometimes this medication is used with aspirin in order to avoid the complications of pregnancy and the so-called repeated implantation failures syndrome, especially when we talk about patients with antiphospholipid syndrome. So it is even used in order to avoid complications of pregnancy in some cases, so it's not so detrimental, it's for sure. But what we know exactly is the fact that actually lupus by itself is a very tricky disease. When we talk about pregnancy development, it may lead to preterm birth, even some gestational problems, low weight of infants, and so on and so on and so on. So, of course, it is better not to use prednisolone before pregnancy, but in case of lupus, it is always lupus by itself, like an untreated state, will be more detrimental to the embryo, oocyte, and pregnancy compared to prednisolone intake. Of course, you have to go on very attentively to the recommendations of your doctor, but still, don't worry. There are plenty of pregnancies that are occurred on therapy for the lupus, and they have usually good prognosis and even no lupus flare. So I hope that everything will go fine, but just be very attentive. Thank you.
Thank you, Olga. Let's take a look at the other one. I want to have a boy, and can the embryologist choose the sperm that carries Y chromosome and to use it to do the ICSI?
Well, if you talk about standard embryologic technologies, no, unless this embryologists have some, special knowledge, but I don't think so. So when we do standard ICSI, we just see the morphology of the sperm, and of course, it doesn't reflect its sex. So there are some techniques that help to sort the spermatozoa according to their sex chromosome, whether it's Y or X. But in Ukraine, these technologies are not used because they are not very popular, they are not very distinct. It means that even if you look for the Y spermatozoa, you have a 20% chance to get actually a girl. So it's not very precise technology, and moreover, you still have to confirm the sex of the embryo that you got with the pre-implantation genetic testing to be sure about its sex. In Ukraine, the pre-implantation genetic testing for only for sex selection is prohibited. It means that you are not able to test embryo only for X chromosome or Y and to know whether it's a girl or a boy and then transfer. Of course, it's prohibited, but you may go for a standard PGT and to get information about not only the sex of the embryo, but about its health too. So when we talk about sex selection of embryos, it's always better to go for a PGT and to transfer the embryo of a sex you are thinking about. Because for assisted reproductive technologies, a pregnancy with a boy is not a case. A case is a healthy pregnancy with a boy. So I hope that would be your future. Thank you.
Thank you. The next one is, what do you prefer, IVF or ICSI? Last time I did IVF because my doctor said that my eggs are fragile. What is your opinion?
Well, basically, we are going for ICSI in our clinic, as a majority of IVF laboratories in Ukraine and I would say in the world too, because we have a high number of patients with male fertility, very severe male factor that are just cannot go for IVF, standard IVF, and of course we have a high number of cycles where we work with a limited number of oocytes due to advanced maternal age. So when we have only 1 or 2 or 3 oocytes, of course we won't go for IVF, because in such cases you always have a risk of so-called total fertilization failure. When the spermatozoa are not able to fertilize this particular oocyte in laboratory, and so the patient stops the cycle just the next day after fertilization. So of course it's a very devastating situation. That's why our standard technique is ICSI. But of course for the patients who have a history of I would say difficult ICSI due to very fragile membrane, because sometimes when the oocytes are very sensitive, very fragile, the glass needle that is very thin still can harm them during fertilization. And then in such situations, we recommend using standard conventional IVF for the subsequent cycles. And if your doctor recommends you to go for conventional IVF, it is of course recommended to go for his recommendation and to do it. Because of course, sperm by itself is more tender to the oocyte when we talk about fertilization. But of course, even if you go for conventional IVF, it doesn't guarantee us the fertilization, the subsequent development of embryos. And of course, it can help to avoid oocyte degeneration, because sometimes oocytes just go for apoptosis without ICSI or with it. So probably in your particular case, knowing your anamnesis, it is wiser to go for conventional IVF for a second attempt. And of course, you have to be very attentive and inform your doctor before the start of stimulation cycle to plan the fertilization in the right way. Thank you.
Mm-hmm. Thank you for your detailed answer. I also wanted to remind you that we have an active chat and you can keep writing your questions there, and we will answer them at the end of our presentation. And we have some more questions. Can I freeze my eggs and my husband's sperm to send them to your clinic? I want the embryo to be cultivated in your clinic, and I also want to do the PGD. Can this be done? Do you think it's a good idea?
Yes, it can be done. Of course, you can freeze your eggs and your husband's sperm and feed them to our clinic, and we will be happy to breed them. But as an alternative option, I'd like to suggest you to go for IVF cycle in your country and to freeze embryos and ship embryos. Because basically, first of all, our sites are more sensitive to freezing process, and if the laboratory has a limited experience of oocyte freezing, the result may be a little bit disappointing. And from the other point of view, the embryo they're just more potential compared to oocytes, because oocytes, they have to go for IVF, they have to go for the first cleavage and go on. While the embryo, when you talk about 10 oocytes, you have you talk about 10 perspectives. Yes, about future perspective. When you talk about 10 embryos, you talk about 10 embryos. I talk about it because usually patients think that if they have 15 oocytes, they for sure will have 15 embryos, and it's not a true story, unfortunately. So if it's a favorable situation, you can go on for IVF cycle, freeze embryos on day 3 or day 5, depending on your particular case, taking into account your number of oocytes, IVF level in your country, and so on, so on, so on. It can be discussed individually. And then ship embryos, it will cost you totally the same, but it will give for more opportunity to us and of course to you in the future.
Thank you. Let's turn to the next question. My family has hearing loss history. Can I check hearing loss gene before I transfer the embryos?
Generally, yes, but in order to test any mutation, it is, by the way, this technology for testing such mutation is called monogenic disease testing, or PGTM. So we can test embryos not only for so-called chromosomal abnormalities, that is the big flaws of chromosomes, of genetic material, but also for a very small. But there is a very important limitation. In order to look for a mutation, we need to know this mutation. So if you have genetic testing and you also already identified the mutation that caused the hearing loss, of course, it's not a problem. Technically, you just go for IVF cycle, do biopsy, freeze embryos. This biopsy is sent to genetic laboratory along with the blood samples of you and, for example, your husband, your close relative with such a hearing loss problem, and according to these blood test results, so-called PGTM setup is done, and a particular mutation of gene may be detected in your embryos. So technically, that's not a problem, but first of all, the mutation should be identified, and we should look for this mutation. By the way, if you will search for the hearing loss mutation, you also can do a standard NGS testing for chromosomal abnormality. Today, thanks God, we have such a proficient technologies that can combine these difficult tests with only one biopsy of embryos. Thank you.
Thank you very much. Sure was very informative. I'm 42 and I don't want to do egg donation. Are there any technologies that can help me to increase the quality of my eggs?
Well, unfortunately, today there is no technology except magic that can increase our quality of eggs. But there are some very, I would say, simple recommendations that are really difficult to go on. It's lifestyle correction. It is some medication testing. I mean, you have to consult your gynecologist to see whether you probably take some medication that may influence the quality of oocytes. And of course, you have to consult a very good specialist who will be able to tell you about the future strategy. We cannot influence the quality and even quantity of oocytes directly, but what we can do, we can go for a healthy lifestyle, be conscious, yes, and then go for so-called accumulative cycle. It means that you can stimulate for several times with freezing effects, and afterwards do their fertilization simultaneously. That will decrease the cost of cycle and increase the possibility to get the healthy embryo and, of course, a pregnancy. But unfortunately, quality is not influenceable.
I see. Thank you very much for your answer. And we have a last question. I want to do egg donation and use frozen eggs. If I use 15 eggs, how many embryos can I have? How many embryos usually pass the NGS? I think this is a question about statistics and what we usually can expect when we do that.
Talking about statistics, we need to keep in mind that statistics always work on a great amount of material, yes, but in your individual case, something may differ, but we will use a very simple mathematics. We will think about the minimal number, yes? Okay, if you have 15 eggs, that with a great probability you will have 12 zygotes, it means fertilized oocytes. Now, these 12 zygotes, at least 6 will develop 2 blastocysts or freezing. If you go for NGS testing, we can expect that at least half of the blastocysts will be healthy. So if we use 15 eggs, then we can at least expect 3 healthy embryos for the transfer. As for me, taking into consideration that is the lower limit, it's quite a good prognosis. So the only thing that I can is just to wish you the bigger numbers and of course a good result of treatment. Thank you.
Thank you very much. It was the end of the questions that we had, but we still have a very long question in our chat, and I think it's a good time to answer them. Okay, so the first one is, do you do infectious diseases testing on frozen sperm? If so, is it testing in-house or at another lab? How long does it take to get the results?
Ooh, it's a very special question, I would say. We can provide disease testing. Of course, it is important to say what infections we are looking for, because of course I think it's like sexually transmitted disease question, but of course we can do it. We have our genetics lab, so we can do standard PCR to look for some most common diseases. But if we talk about I don't know, some strange ones like maybe African parasites or something like that. So we can, of course, go for another lab. But of course, if you talk about STD, it's our in-house testing.
Okay. The next question that we have in our chat is, we have had testing on our embryos at only tested 9 embryos. We have more, but didn't test them due to the cost. 8 out of the 9 embryos were very good. The daughter is healthy, young, 23 years old woman with a healthy young child. My husband's sperm was very good. We had DNA fragmentation on his sperm and results was good to excellent. We are worried about transferring 2 embryos due to potential risks of twins. So thinking about using 2 surrogate mothers. What are your thoughts regarding the birth of healthy babies? I'm thinking of transferring 2 eggs of 4AB. Have you received any hand-carried cryo shipments during the latest border closing in Ukraine, which started from 31st August a week ago? Would there be a possibility that my courier will be stopped at the airport, not allowed in? This is more of organizational questions, I think. And we have one more. Even though we did NGS 24 testing and we have good embryos from our donor and my husband's sperm, as per my above question, is there a possibility that either her or my husband's carrier conditions might come out in the babies, even though they do not carry any of the same carrier conditions? It's pretty long, so maybe we should try answering the first part first. Okay, please, Olga.
I will be honest with you. I always prefer transfer of only 1 embryo, because naturally we are done to give birth only to 1 fetus. When we talk about twins, we have higher possibility of complication, preterm birth, maternal complications, and of course, it's not a very good prognosis. From the other point of view, you can see plenty of twins that are healthy, they're happy, and where pregnancy outcome was just perfect. So I would say it's not my right to recommend you as a personally what is better, but from the professional side, as an embryologist, of course, I would insist on transferring only one embryo to one surrogate in order to avoid any complications, because when we talk about transferring two embryos, of course, we increase the possibility of pregnancy occurrence, but from the other point of view, we almost 2-3 times increase the possibility of complications, birth complications. So it's not the risk that we would like to take. If you have a possibility to do transfer to 2 surrogates, it is a very good option, and I would support you. I think I'm done.
Yeah. I don't know if you can answer, like, if we have received any hand carry cryo shipments during the latest border closing, or maybe it's the organizational question that's better not to be directed at you.
Well, as I recall, we received, yes, from the 31st, and we actually will probably receive today.
Okay.
We are waiting for the delivery.
Okay, great. If you later need any more detailed information, you can contact one of our team, our coordinators, and they will give you more detailed answer about the organizational questions. We have one more message. Yes, it is sexually infectious diseases test because my lab in Australia didn't do it because his wife, I would have received them for carrying pregnancy, and I'm the wife, and they don't test wives here. We've done 12 IVF cycles, and never I or my husband have ever had any sexual infectious diseases. This would probably be piece of
I got it. It's about the first question. Well, it's difficult for me to read in such a position, so I beg your pardon for my strange pose, but just want to understand the question fully. So I see. Well, of course, we can go for such a testing. That's not a problem, but actually we can talk about, now I'm trying to answer this question about sperm testing. Actually, if your husband had frozen sperm, yes, you may go on for not testing a sperm. Why? Actually, he can go for blood testing of sexually transmitted diseases. We're not looking for like very rare cases, yes, like strange conditions. We are looking for very common diseases like HIV or syphilis. So you can go for blood testing, and probably, I don't know, we can discuss it with a doctor, but as I see, if your sperm was frozen and afterwards, like in some time, you got the testing of a blood, it automatically, and you're clear. It means that automatically the sperm is also clean. So no need to test sperm by itself. Blood is quite enough. And I don't know what's the well, I think that we need to think this situation individually. Like, will it work, or as I see, the sperm was frozen some time ago, and it's really difficult to repeat freezing for some reason. So probably we need to think about it. How would be better to test sperm, for example, here, and how we can do it, or maybe it would be better if it's possible to refreeze the sperm with blood testing. Of course, I see that probably you have some reason why it's not an option for you, but still we can think about a option that is the most comfortable and the most efficient for your case. And also the answer to NGS testing, yes, and I see. Well, I think that if you contact our international department, and of course we already thought about this situation, probably we can think about the possibility how to do it the most efficient way, okay? You just have to text our beautiful colleagues and very clever colleagues, and I'm sure that we can solve this situation without any problems.
Yeah,
and also
work individually in that. So you know that you have linked our website. There's a contact email there. There's a phone there, so there's no problem getting in contact with one of our coordinators, and we will make sure to deal with your case in the best way. And if there will be a need of this, we will of course organize also a conversation with our embryology team, with our doctors, so that we can think of the best solution for you on the matter. Let's take the last questions that we have already here. I have embryos, but sending extra sperm, which we have many straws of, how long do the results take? Let's take this one and we'll be finishing. Please, Olga.
Well, I think about it's the second part of the question about STD testing. Well, it will depend on the facts what we actually will test, but the result will be in a couple of days. Usually STD testing doesn't take a long time. And we didn't answer also the question about carrying some disease. Unfortunately, even if you do NGS, you do not screen embryos for some monogenic diseases. But if you go through the carrier screening and your donor went through the carrier screening, I mean, your husband went through the carrier screening and the donor of oocytes also did, or you did, I don't, donor. So if the donor did the carrier screening and the husband also did, we have a very, very tiny chance of such bad situation. Basically, carrier screening goes for the most common monogenic diseases, and if you didn't find any risks about it, then probably we can sign and think that everything will be fine. Of course, there's a very, I would say, like, it's so tiny chance. I prefer to be honest. It is always a tiny chance for some problem when we talk about genetics. We cannot be 100% sure that it will not arise, but we can be sure for 99.99999. And I think it is your case, and I hope that everything will be fine, because as I see, you did almost everything you could to to make the situation safe.
Okay, thank you very much. I hope.
No. It's also a question about surrogate and epigenetically switching of any diseases. No, if the embryo has any mutated gene, well, it can work in any conditions. But if it doesn't have, then probably it won't be influenced by, we have such a lot of conditions in our life that can influence our embryologic development. So it is sometimes surprisingly how we can even get birth. But it is I don't think, no, it's not a risk actually. If the embryo is healthy, it will be healthy, and the only thing that we can recommend is to healthy lifestyle for the beautiful baby.
One more question.
No, I don't think that surrogates are genetically tested because they are, as you already said, their genes are not involved and they would not, even if they are carrier of any mutated gene, it will not anyhow influence the development of embryo. And if they have some disease, of course, we would know that and they would not be tested and go for surrogacy. So it's not a case they should think about.
What
Pronuclear spindle transfer? Yes, if you talk about pronuclear and spindle transfer, then in such situation we have to test the of course, if you talk about pronuclear and spindle transfer, it's a very, very sensitive question. Of course, we, in such case, work with your genes. So we have to test you and your husband if you would like to, if you don't want to support, to rely on nature, yes, and you would like to test, then first of all, you have to test your husband and yourself for carrier screening. I think that that was the last question, and there was a lot, and that was interesting. Thank you for your active.
Yeah, it was very informational. We had a little of a case study, so we really got the opportunity to discuss, I think, at length the problems of one of the couples that are facing. So if there are not going to be any more questions in our chat, then I think we should thank our speaker, Olga. Thank you very much for being with us today and for going in such long length and explaining everything in such detail. I'm sure it was very informative. If you have more questions, just contact one of the coordinators on our team and we will make sure to get answers for you. If you missed some of the parts of the webinar, or you would like to watch them and listen to them one more time, you'll be able to find them on our YouTube channel. And make sure to join us for the next webinar next week. Thank you very much.
Thank you.
Goodbye.
Take care. Bye-bye.